be talking to us about COVID-19 and genomics and proteomics research in Chilean patients. But before we get to hear the talk, a little bit of an introduction. Dr. Verdugo started in his career and training as a veterinary medic with the degree from University of Chile in 2001, and then went on to do a PhD in genetics at the University of California, Davis, which he completed in 2007. He was working on fine mapping of QTLs for obesity in mice, and then continued with a postdoc in computational biology at the Jackson Laboratories with Dr. Gary Churchill. At that point in time, looking at statistical analysis of transcriptomic data from mouse crosses to inference gene networks underlying complex traits. His second postdoc was at INSERM, where he applied systems genetics to human population studies of cardiovascular disease. And for the past 10 years, has been an assistant professor at the Human Genetics Program at the University of Chile. He is the director of the Chile Genomic projects, looking at genetic diversity in the Chilean population and the relevance in health, directs the genomics and the bioinformatics laboratory, and leads the UGenoma, the national academic network, which is not just to support academics, but also at graduate level education, both in genetics, genomics, and bioinformatics, and really leads on the GenoNet multicentric COVID-19 cohort. He is currently at the University of Talca in Chile, and it's a great pleasure to welcome Dr. Verdugo to this webinar. So very much appreciate your time today and looking forward to your talk.
Thank you very much, Alex, for that kind introduction. And I apologize that I haven't updated the first slide with my affiliation, but as Alex mentioned, I just started a new position at the University of Talca, though for the moment I have a double appointment with both with the University of Chile and the University of Talca. So today I'm going to present to you the design and the motivation and the results of our COVID-19 cohort that we have established since 2020, and that we are currently following up for in looking for long-term symptoms of or sequelae of this disease. And I'm part of the COVID-19 Genomics Network, or C19 GenoNet, that have provided much of the samples and work that I will present to you. Well, as you must know, there have been a number of studies worldwide to look for genetic variants for both predisposition or severity of the disease, and this is probably the largest one, published in 2020 by 23andMe, by taking advantage of the large base of participants in their company, in their services, when they reported an association with the ABO locus. That is well known, but also a higher predisposition in Latino and African American participants that could not be explained by socioeconomic status alone, which suggests that there are some genetic variants that are ethnic or ancestry specific that may hold relevant information that cannot be explored if we only study Europeans. And that's why we established, well, previously the Chile Genomic Cohort, which is just a survey of the general population that provided us with some baseline data that could be used for preliminary studies. And in total, we had over 3,000 people in this. This is not a cohort that is being followed up. It was just a transversal study at that time, around 2013, but that captured a lot of the genetic variation that is present in our population. And using that information, we went ahead and test whether we could find any variation in terms of polygenic risk scores for the associations that had been published already. And the susceptibility PRS was calculated based on the study by 23andMe, and the severity by Pyro Castaneda, which is the Genomic network, which I'll talk a little bit more later. And we did find data for those SNPs in our datasets, which are marked here with a check mark. Although this was microarray data using the Axiom, Affymetrix Axiom Latino array, which we then imputed using TopMed. And what we found is that for the lead SNP in the ABO locus, the allelic frequency for the SNP is very different between ancestries. As you can see, the risk allele, which is associated with the A group, is much higher in Europeans. It's almost absent in Native Americans. From this part of the continent, which the most common one, ethnicities here are the Aymara in the north and the Mapuche in the south. And Chileans, which are here ordered from north to south in different regions, you can see how we have an intermediate level allele frequencies for this risk allele. And if we calculate a susceptibility polygenic risk score with all of the SNP published in that paper from 23andMe, then you see a very similar pattern, but this is driven mostly by this lead SNP. Now for the severity phenotype or polygenic risk score, we see the lead SNP is in chromosome 3. This association has been repeatedly reported in multiple studies, and we can see how the relationship now is inverse, where the Mapuche have the highest allele frequency than Europeans, or even it's not seen in Africa. This is just a sample of 30 individuals from the 1000 Genomes project. You can see how it's not present in those ancestry. And in the Chileans, it's similar to the Mapuches. Now, if we calculate a polygenic risk score with 8 SNPs taken from the genomic study, we see that the polygenic risk score is higher in the Amerindian ancestry, and it's intermediate between the Europeans and the Amerindians in the admixed Chilean population. So this is to show that even for those SNPs that have been reported in the literature in European population, we see differences in allelic frequencies that explain or predict that the population will be at different risk levels. Of course, this can be affected by many factors. There are the pattern of linkage disequilibrium can be different between different populations, so we cannot assume that the actual level of risk will be equivalent to what the pattern we see here among populations. And that's why we have to confirm this with actual case control studies, and that's what motivated our study. From this part, we observed that the Chilean population has an intermediate allele frequency for both the susceptibility and the risk alleles, although in different relationships, and that a polygenic risk score for the disease may be lower in people with higher Amerindian ancestry, and that Amerindian ancestry may be associated with increased risk for severity forms. However, that has to be confirmed, as I said, with local case control studies. And that's how we were granted, we designed a study to do this in Chilean patients, and this was granted in 2021. And it's a work that is a joint effort between multiple universities from the north. University of Tarapacá and Antofagasta are in the far north of Chile. Then University of Atacama, also in the north, and University of Chile in the center, University Austral in the south, and Universidad de Magallanes in the extreme south in Punta Arenas. So we covered the whole geography, all latitudes of Chile in this study. And our design was to recruit 4,000 patients, regardless of their level of severity, to apply a survey, to create a biorepository that will be stored in the institutional biobank so that it will be available for everyone to use, to genotype everyone using microarrays, and to perform local GWASs. So we did an open call in different media outlets, in radio, newspapers, and so that everyone will be invited to participate. We had about 3,000 people that contacted us through the different channels that were interested in participating, and so we had a great reception, and everyone was motivated to participate and help in any way they could to better understand this disease. But since we had a space for even more participation, we did some work with local hospitals and also some testing companies to go actively and invite participants in controlled PCR testing that was done in different companies or among patients of hospitals. And here you can see some pictures of the team. Here's the team at the institutional biobank, where they prepare the recruitment packets, which was very important in order to have exactly the same recruitment protocols across all the different centers. Everyone was pre-labeled, pre-printed the survey, the barcode for the participants and the tubes, so that we were very rigorous in terms of making sure that this exact same protocol was used across all the different recruitment centers. And also you can see some pictures of participants in Magallanes, in the extreme south. As you can see, very happy to be part of this project. So the recruitment, as I said, was through public announcements and active recruitment, and also clinical lists in participating hospitals. And regardless of how they got into the project, we made an appointment for a visit where we invite people to come to one of our recruitment centers, and in some cases we also go to their own home address, perform the process of informed consent, an interview, and depending whether the person was hospitalized or not. If the person was hospitalized, we captured the information in a clinical record form from the health record. If the person was not hospitalized, then we captured through the survey into the standardized form in REDCap. All of the information is in REDCap, and our bioinformatics team was routinely every week performing automatic checks on the quality of the data, looking for duplicates, for missing value, for outliers, and that was reported back to each of the teams in the different recruitment nodes, so that they will go ahead and check whether they could correct the data based on the printed forms or eliminate duplicates and so on. And if it was necessary, we recontacted participants, because we had the email and the phone number, so that we could check some information that may not be correct. So that was a long process of data curation and completion that was very, very important in order to have good quality data in the database. In terms of the sample, we drew a blood sample from which we created different aliquots. So we have stored plasma, buffy codes, and DNA. And everything was done using standardized protocols that were previously validated in the biobank, and the whole process, even the times at which each of the steps were performed, was recorded and was monitored in real time from the biobanks, so that we could correct any deviations from the protocols that may have occurred. And as a result, we see here some statistics about the biorepository. So because we have 4 plasma tubes per participant, half of the biorepository is made of plasma samples. As you can see here, the proportions are similar, of course, across the different centers, but the number of participants is different across centers because they started at different times. The first one was actually the Hospital del Salvador in Santiago, but Magallanes was very prolific in terms of recruitment participants and had the highest number of samples. And these are ordered from north to south. In terms of effectiveness or completion, so the gray means that we have all tubes from a participant, and red that we have no samples, or in yellow that we have some samples. So as you can see, for 90% of participants, we have all tubes, and only 7.8 we are missing some. In terms of DNA concentration, which was recorded during at each site during the processing, here we have the minimum that was established by the genotyping core, and you can see for the large majority we are above that minimum. In some cases we had problems that we were able to later correct by doing further training and adjustments to the protocol according to the local availability of the different types of equipment and expertise of personnel. In terms of spread measures, which are common in biobank to determine the quality of the processes, which relates to the time that goes between collection and centrifugation. The ideal is that there is no more than 2 hours between data collection, sample collection and centrifuge. And that will be in dark green here, which was the case for 40% of samples. And for the rest, it was mostly under 4 hours, which is very good for biobank standards. So we are very happy with the speed in which samples were processed, because that ensures that the samples will be collected not just for with enough material and enough quality for one study, but that will be good for many studies in the future. In terms of the survey, we established different severity levels from asymptomatic to lethal, and you must be very familiar with this. So the mild it's people that did not require hospitalization, and then hospitalized could be just hospitalized or hospitalized severe, which means that they needed some oxygen but without mechanic ventilation, and critically ill, well, those that require mechanic ventilation that were that had to be accepted into an intensive care unit or similar, and of course the last level is lethal. And in the survey we collected demographics, ancestry, socioeconomic status, different comorbidities, and in terms of chronic diseases, and 26 different symptoms, some records about the healthcare path of the person, and some long-term effects. This is a progression of the recruitment from late 2020 to the beginning of 2021, and this was previous. The whole sampling was previous to the start of the vaccination campaign in Chile, so all of our samples are previous to vaccination. And this is the distribution of severity. As you can see, most of our participants were mild, And that relates to the fact that we did an open call, and most of the hospitalized and severe cases came from our relation with some hospitals to include the hospitalized patients. And this is the distribution by severity and geography across Chile. As you can see, the country is divided in two halves here, the north in the left and the south in the right. The size of the dot here, or the circle is associated with the number of samples. In terms of ancestry, as expected for every Latin American population, you can see that in the first PC, which separates Africans from the rest, we are very much on the left side, because in Chile there is little African ancestry. We did not exclude individuals by nationality, so we do have about 35 foreigners, and those represent these individuals with the highest levels of African ancestry, and which we exclude from analysis. And in the PC2, which differentiates the East Asia or versus Europeans, we Chileans are across that along that edge, depending on the level of admixture between Amerindian ancestry, which is more related to East Asian, and the European ancestry related to our Spanish ancestry. So this is very much as expected for admixed populations, and it tells us that we must include admixture in our analysis. And some results we have tested now the PRS calculated based on our data. The samples, the DNA samples were genotyped using the Illumina GSA array, and then imputed with TopMed. And after that, we calculated the PRS using the critical illness SNPs from the Omics paper. And what you can see here is that, well, ancestry, Amerindian ancestry, has a positive odds ratio, meaning that the person with 100% Amerindian ancestry has almost 5, odds ratio of almost 5, versus a person with 0 Amerindian ancestry in our sample. And that was significant. And whereas the polygenic risk score has an odds ratio of 1.8, which was very significant. And that means that per every unit of increase in polygenic risk score, which is in logarithm scale, we have this level of increased risk. And now if we test for interactions, the two-way interactions were not significant, but the three-way interaction was significant with sex and Amerindian ancestry, where we have a 218 odds ratio for a particular combination of high Amerindian and sex being women, and high polygenic risk score. We can be seen in this slide where people were divided by 3 terciles of Amerindian ancestry and 4 quartiles of polygenic risk score, which we call polygenic risk level. So 4 levels of polygenic risk score. And you can see here in gray the controls, and in dark gray or almost black the cases. The proportion of cases increases with polygenic risk level, but particularly so in women, and that trend is higher in the women with in the highest level of Amerindian ancestry, okay, which is this level here. This is where we see the highest trend, although the trend is also present in men, but it's higher in women. So that was an interesting finding, because we were expecting to have a higher trend for people with higher level of European ancestry as opposed to Amerindian ancestry. because the polygenic risk score was constructed based on SNPs that were associated in a European population. But we see the reverse associated with higher allele frequency of those SNPs in the genomes with Amerindian ancestry. Now, so that was the work done by a student, Laura Carvajal, and now the results for another student, Juan Saez, who is a computer scientist. He wanted to explore whether we could use both the clinical and the genetic information to predict severity in patients, in Chilean patients. So these are the results of different modeling that he did that can be seen here, like random forest, logistic regression, decision trees, XGBoost, support vector machines, in order that will consider all of the variables that we included in our survey. And you can see here how there are some variables that are consistently being associated with the as being predictive of severity. And these are in common with the reports from others like obesity, vitamins, drugs, and so on. Even self-reported anemia. Okay. Here are the ROC curves for using this for using one of these methods, XGBoost, only with clinical data. We get a area under the curve of about 0.85, which is pretty high, I believe, for this type of data, in order to discriminate people that it will be considered critically ill versus not critically ill. And now he went ahead and moved forward in order to include the genetic data using what is called neural networks. He used a multi-layer neural network that will be capturing the information across all of the SNPs into this intermediate level variables that are constructed with this method in order to finally discriminate between one case or the other being critically ill versus not critically ill. And he tested this by including the genetic variants only that had been reported as suggestive of association genome-wide. It did not work well with millions of variants, or at least we could not solve that in a reasonable amount of time. So we did this based on only the suggestive level variants. And what you can see here is that the F1 score that weighs both the accuracy and recall of the classification for critical illness was improving as you include more genetic variants, but up to a point, and then it starts decreasing, just adding more noise. If we zoom in this region where there is a gain from including genetic data, we conclude that including about 100 or up to 200 variants is beneficial for a prediction, and that is way more than the number of variants that have been reported with a genome-wide significance as association for critical illness. So that means that there is valuable information beyond those few variants that have been statistically associated, and that should be explored for their level of association of prediction for this phenotype. And finally, we participate in the international effort to map variants for this disease. So far in the published studies, Chile was not present, but now we are present in the latest release number 7. This is not release number 7, this is the previous one. You may be familiar with this, but yeah, this is the previous one, but as a comparison for the C2 phenotype, which is being a case versus being part of the population, using a population sample as controls. There are a number of loci that have been associated by this international initiative, being led by the ABO locus, as I mentioned before, but also loci in chromosome 3. Of those, we did not see an association. We only see an association in chromosome 6, which, but not in the same locus as reported by the international organization. And we think that it's very relevant that the ABO is not associated in our population. And actually, by collaboration, I don't know if I have a slide for that, but there is a similar effort in Mexico, and also they don't find an association in ABO. And we believe because that the frequency of the alleles in the ABO locus is very different between the Europeans and the Amerindians. So although the A allele may be associated in the Europeans, for some reason that either because the allele is at very low frequency in our population, or some other local rearrangement, we don't see an association here in this part of the world. For the B2, which is being hospitalized versus being member of the population, we don't have that many cases in this. But this is a preliminary study, so we have a better one now here for B1. In B1, we have more controls because we had a lot of mild cases, so we have better power for B1, and you can see how we replicate very well the chromosome 3 locus at the LZTFL1 gene, which was earlier this year in January. There was a paper confirming the functional effect of this variant in the binding region of zinc fingerprint transcription factor that affects the expression of the gene. So this has been validated, and we do see that association at the genome-wide significance level in our sample, even though we don't have that many cases, just 512 cases. In the A1, which is the critically ill phenotype, interestingly, we have the same locus as in chromosome 3, but we also have a second locus that is almost at the significance level in chromosome 13, which were not previously reported in any other study, except just very recently in the past weeks. The latest version, release 7 of the International Association, that does include our data, and among others, Mexicans and other latitudes, they do have now a locus, an association at the end of chromosome 13. Although I must say that it's not exactly the same as ours, but it's very close. Just the peak is not overlapping the same gene in both results. That's very interesting, and we are exploring on the reason why we have different signals with the international initiative. And in comparison with the genomic study, which is the largest one for critical illness, there was no signal in chromosome 13 at that position. So this was only based on European participants. So we think that there is an association that is particularly for Amerindian ancestry here. This is a little bit of the same. So at the end, I wanted to mention the C19 GenoNet or the COVID-19 genomics network that we have created from our group in collaboration with other similar projects that were funded at the same time in Chile through a call that was created by our national funding agency because of the pandemic, and about 60 different projects were funded to study COVID-19 at the same time, and we saw that that was a great opportunity to work together to a common objective, and we seek to establish common protocols and objectives so that we could later put our data and samples together in a common resource that could be used and shared with the community. In our case, our objective was to now include information for the virus and the microbiome of patients to be analyzed jointly with the genetic data on the host that we already had generated, and also to study the genetics of reinfection and long COVID. So we partner with a number of institutions along the country to do this and establish a structure that will ensure that we continue working together with international organizations such as the IHCC, which was one of the partners that provided us a letter of support for this project. So we are very thankful, along with ICDA and others that had believed in this project. And also we paid a lot of attention to the training of young researchers here in Chile that will be part of this and that will benefit from also from our international collaborations in their training. We summarize here the types of samples that we have, the type of data that we have in the different centers and different projects that are all available for this study and for the biorepository. We total over 3,000 individuals with data and samples stored in the institutional biobank, which is located in the University of Chile, but we are also in the process of creating a national network of biobanks that are connected, that have the same protocols, and we have hired services of a company that will help us to develop a catalog that will be online so that people can find the samples that are located in any of these nodes. Hopefully in following in the next presentation, I will show you that working already. And I would like to finish by acknowledging the work of many people. I'm showing just a few here, the heads of the different groups, but this is the result of a lot of groups that will be meeting weekly during the pandemic from our homes. We will learn how to use Zoom together by doing this study, and we are happy that finally we are able to present to you some of our first results, and we are working on getting this data published soon. Thank you very much, and I'm happy to take any questions.
Thank you very much for a great talk and a lot of effort and I guess coordination of all the sites across Chile. And I was actually really quite impressed that even though you had, I think, the protocol to have the samples under 2 hours, anybody having collected samples from many participants in a hospital setting or even the community, to have those, or a majority of them, under 4 hours, that's quite an effort. So must have been quite a coordination, quite an I guess countrywide effort. So I don't know whether anybody has any questions, but I just wanted to ask, maybe start with a question on the PRS score and how many SNPs were included. So I know you have talked about the chromosome 3 and the ABO locus, and then later on chromosome 13, but sort of how many did you start with?
With the 8 that were reported in the genomic study, the 8 SNPs.
8 SNPs, okay. Yes. And the second question, I think that your student who was, I guess, a computer modeler and looking at the different ways of using the survey, the epidemiological data, it seems that I think the support vector machine had picked up the other medication as one of the key, I guess, variables. Could this be because these were patients at risk are those that had other comorbidities and they were already taking another medication? Was it a proxy for somebody who was already unwell and more prone to getting COVID-19?
I believe that the variable that was selected there, I'm looking at this was no drugs. Oh yeah, medication, sure, yes. So so we have both medications and comorbidities in the modeling. So so in theory they could be picking different associations there because both are in the modeling. But of course it could be related to this as well. We cannot discard it.
Okay, great. Does anybody have any questions? I can see there is a question in the chat. I think Scott, maybe you can.
I should raise my hand. Ricardo, I had a question about your continued monitoring of these cohorts. And you mentioned long COVID. So that's good. But I'm also interested in the types of phenotypes you're monitoring. Is there anything that relates to mental health, anxiety, depression, suicide, those sorts of information? Are you collecting anything like that?
Yes. We actually, I can show you the form, and we can share that with the membership as well. The forms that we are using, we are in the process now of finishing our first recontact after 1 year, and in that we do ask about mental health. And also impact on the daily life activities at work, at home, but it's not a mini mental type of, it's not that deep into one particular set of phenotypes. So what we think, and this, we are in collaboration, working together with other cohorts in Latin America now, in Costa Rica, Mexico, and Brazil, and the researchers in Costa Rica are particularly interested in those type of phenotypes because they are psychiatrists. And what we think is that we are gonna later recontact people with a mini mental survey. But if there are other similar efforts in other members of IHCC, that will, I think that it's a good time for us to have that discussion and define exactly how to do that, because what we didn't want to do is to have a very lengthy survey that will try to capture every possible phenotype now in the first recontact, because it will take us a long time and people may not want to be with us for so long. We are also interested in metabolic phenotypes and physical activity phenotypes. So that's why we do have mental activity, but it's not very, very deep. So we'll be happy to discuss with the specialists on that, whether there is a need for if you guys are applying another survey in another cohort and we would like to have the same type of data across cohorts, then we have the opportunity to contact people again, either by email or by phone.
I will send you some information. One of our IHCC pilot projects was on mental health related to COVID, and it's investigators are Jordan Smoller in the US and Sarah Bauermeister in the UK. And they do have a number of various countries. I want to say Brazil. I think Andre Brunoni is collaborating with them from Brazil. But I know they're always looking for other COVID cohorts to include. And if you don't have exactly the perfect mental health phenotype, maybe there could be some in between. This is what you have right now, but this may be the type of questions you want to ask in your case report forms going forward.
Sure. Yeah, I'll be happy to talk to them, send them what we have, and then go from there.
Yeah, fascinating results too, especially the Amerindian result.
Thank you.
Alex, there is a question on the chat box by Catherine Marceline. You are muted, Alex.
Sorry, I'm on mute. Thank you very much, Ricardo. There's another question in the chat saying, did you find association of severity and the recruiting site, whether at region, county, or hospital level?
So as you can see in the map, I'm going to. Where is the map? Okay, here is. So yes, we have divided the country in different ways. The most sensible one is by macro zones, which are far north, near north, center, south, and extreme south, and so that we have enough number of samples. Oh, sorry, these are just the. No, it's okay. And we do see differences, but that's related to the fact that we did not partner with hospitals in every site. Therefore, we have more representation of critical illness in the center and of the country and than in other places. So we do control for that, but it's not an association of interest because of the sampling could not be done perfectly so that that variable could be included. Okay. And that's the reason. But yes, there are differences because of that. So we try to therefore control for that, but focus on the association with ancestry, PRS, and other clinical variables.
Thank you. And I think seeing that again, it's very, very helpful for that question. Does anybody else have any questions? I have a comment from Ronglingly saying that it's an excellent presentation. So
thank you, Rongling. Good to see you.
Yeah, I also need to have a comment about your work. Since your cohort has done an excellent job, and maybe some other cohort within the IHCC have also do some work similar to yours, maybe we can connect those cohorts who have done something about this, and see if they can do some meta-analysis or some other analysis to really leverage the IHCC these advantages of
here in Chile or somewhere else?
Oh, somewhere else.
Okay.
Yeah, I mean,
sure.
Yeah, since IHCC have so many cohorts, I'm sure that other cohorts probably also do the same thing.
I would love to do that, sure. And actually, what we have done is we got in contact with one of the members of IHCC here in Chile, the Mauco cohort, and through a partnership with CogNet, which is an international network funded by NCI at the US. They did genotype. Well, they are in the process of genotyping 1,500 people among the cases of the Mauco cohort. So we are already in contact with them, and we plan to use that data as a replication cohort for our findings here within Chile. And also we are collaborating with people in Mexico and Brazil to do a Latin American meta-analysis of our results and see whether we find similarities or differences. But of course, we will be happy to partner to share summary statistics or even data with the other members of IHCC. And this is great of being here so that I can tell you all that we are here, available, and happy to collaborate with other members of IHCC.
That's great. Thank you so much, Ricardo.
And we are in the process of getting this cohort recognized as a member of a cohort member of IHCC. We have submitted the information, and so far it's not among the options when you register for your events as one of the cohorts, but hopefully we will be recognized as a cohort in the near future.
Great. Thank you so much. Nice presentation and the excellent work. Yeah.
Thank you, Ronglin.
Great. Thank you, Ricardo. And Latifa has just put in the link to a talk that happened in January, and she'll send you the recording or the link to the recording separately, so you can connect to those that are working in the mental health and COVID-19. So I think this is, I think the educational webinar series are really now making sure that people between the different cohorts connect and speak to each other, and really, I guess, enhance what we're already learning through consortia, and which we have been for the past 2 decades, I think, across the world, but I think continue to do so through this endeavor. So thank you so much. So if no one has any other questions, I would like to express many thanks for giving your time and for a great presentation. And hopefully, and I think that Latifa has said that you will continue with our panel session, and hopefully hear from you or your cohort, your representatives in the years to come.
Yes, I'll be glad to do that. See you at the panel.
And Latifa, if you have
Yeah, just quickly, I put the information on the chat box about that August we will have no talk. It's going to be summer break, but in September we're going to have the guest speaker is going to be Dr. Nahla Afifi, and her talk is going to be on Qatar Biobank. So please stay tuned. I will send you the dates later. Okay, and thank you again, Dr. Ricardo. And also we will be informing you about the panel discussion on COVID-related cohorts very soon.
Great.
Thank you.
Looking forward to it. Thank you.
Great.
Thanks to everyone for attending.
Bye-bye.
Thanks, everyone.
Bye now.
Bye-bye.
Thank you.